Hello friends and dear students. Today I am going to discuss about chromosomal karyotyping, which is a type in which basically we study the chromosomal structures of any person and detect on the study of chromosomes different types of diseases and diagnose them. So let's start today's lecture. So basically, if we go to the study of chromosomes, or chromosomal karyotyping is a branch of science which comes under the cytogenetics. And it was first discovered in the mid 19th centuries when Carl Wilhelm von Nageli, a Swiss botanist, first observed the plant cell in the year 1842. He observed a thread-like structures in the nuclei of plant cell and named it transitory cytoplasm. In 88, Waldeyer named it chromosome. In 1956, the diploid number of chromosomes were established in humans. Several milestone discoveries in standard cytogenetics are summarized below. In 1931, Barbara McClintock explained the transposition of DNA sequences by analyzing the chromosomal structures of maize chromosomes. So transposition is, we know that other term is jumping genes, and some of the nucleotide sequences, they change their position from one place to another in a chromosomal structure. So it was discovered in 1931 by Barbara McClintock. In 1950, colchicine was used to arrest chromosomal divisions in the metaphase structures. So it is derived from a plant known as colchicium, and it is one of the most important step in the stages of chromosomal karyotyping, which we will see in the coming slides. In 1956, Moorhead and co-workers developed a method called peripheral blood leukocyte culture or karyotyping. So the credit for studying of peripheral blood leukocyte culture goes to the Moorhead and his co-workers. In 1956, J.H. Levin established human chromosomes as 46. So exactly there were different views regarding the chromosomes, how many exact chromosomes are there. But in 1956, J.H. Levin established that it is a total 23 pairs of chromosomes are there, in which 22 are autosomal and one pair is sex chromosomes. In 1959, Lejeune discovered trisomy 21 Down syndrome, means that instead of one pair of chromosome, there is one additional chromosome added to the chromosome number 21, and that such condition is called as the Down syndrome. 1959, again, Ford and co-worker observed the presence of single X chromosome. That is, see, there is a two pair. In female, there is X, and in male is XY. So here it was seen that instead of what two XX chromosomes, there was only the one X was there, and the condition is called as the Turner syndrome. In 1959, Jacobs and Strong observed the presence of one extra chromosome, 46XXY, in humans. That is also called as the Klinefelter syndrome. So, in one part, in one Turner syndrome, one sex chromosome in the pair was deleted, whereas in the Klinefelter syndrome, one extra pair was added in the sex chromosomes. So coming to our step, first we will know what is karyotyping. So the word karyotype is derived from the Greek word kernel, which means content of nucleus. The term karyotype is referred to a group of characteristics that allow the identification of particular chromosome pair on the basis of general morphology of a set of chromosomes at metaphase of a somatic cell of an individual. We know there are different phases in the mitotic phases, G0, Z1 as metaphase, M phase. So here it is the cell cycle the cell stage is arrested at a metaphase stage because it is the best stage to study any of the chromosomes, because chromosomes, all the dividing chromosomes are separate, and it is very becomes quite convenient to study the its morphology. Now, karyotype concept was derived by S. Navashin based on the observation that the number of chromosomes and morphology of each chromosome pair is normally constant and characteristics for a species. So it was the concept that all the chromosomes and morphology is normally constant, and if there is any change, it can be easily detected. Karyotype concept is a phenotypic appearance of a chromosome and its homologous pair, which helps to differentiate types of chromosomes based on their morphology in an individual species. Karyotype is a method of arrangement of pair of homologous chromosomes of a cell in decreasing series of their size. Karyotype study is very important to identify abnormalities in chromosomal structures and numbers. So it studies both the morphological structures of the chromosomes and also its numbers, whether any chromosome is added or it has been deleted, as we have seen previously in the Turner syndrome and Klinefelter syndrome, respectively. Now, karyotype study also helps to determine origin and evolutionary relationship among different taxa. All species are characteristics by a set of chromosomes to carry their genetic information. The chromosomal composition of each species has a number of characteristics. The karyotype is a set of characteristics that identifies and describes a particular set of chromosomes. These characteristics, which are described by karyotype, are the chromosome numbers, relative size of different chromosomes, position of centromere and length of chromosomal arms, presence. Now we see that the central part is the centromere, and one is short arm, that is the P arm, and another is a long arm, that is the Q arm, which is the two parts of the chromosomes. So here also it is easier for us to understand the position of centromere and length of chromosomal arms. Presence of secondary constrictions and satellites, banding pattern of the chromosomes. Basically, it is based on the heterochromatic and euchromatic regions of the chromosomes. The basic of this, where is the heterochromatic, it is AT-rich, and euchromatic is GC-rich chromosomes. Then features of sex chromosome. So what is karyotyping? The process of preparation of karyotype of a species is called karyotyping, that is the preparation of its chromosomal mapping. Basically, we see that all the chromosomes, their structures and numbers. Karyotyping is now most commonly used in clinical diagnosis and clinical genetics. Karyotype is prepared from microphotographs of metaphase chromosomes. The metaphase chromosome is selected because at this stage, the chromosomes will have maximum condensation, that is maximum thickness. At metaphase stage, the chromosome will be visible through an ordinary laboratory microscope. For the clinical karyotyping, the sample materials used may be cells from biopsies, bone marrow cells, blood cells, or cells from amniotic fluid or chorionic villus. So these are some of the specimens which can be used in the process of chromosomal karyotyping. The sample cells were first cultured on artificial medium with suitable growth regulators. Then the cells are arrested at their mitotic metaphase phase by treating with colchicine. Colchicine will arrest the cells at metaphase stage since it prevents the formation of spindle fibers. In the absence of spindle fibers, the metaphase stage cannot proceed to anaphase. Then the cells were fixed with suitable fixative and treated with specific stains to produce characteristics banding patterns in the chromosome. So specific staining or banding techniques are used to identify the homologous pairs of chromosomes within the cells. Cells are then observed through the microscope and the photographs of the microchromosomes were taken. The individual chromosomes are cut out from the microphotographs and then they are lined up by size with their respective patterns to form the karyogram. A uniformly accepted pattern is used for the arrangement of chromosome in the preparation of karyogram. Is that in a karyotype, the chromosome of the organisms are ordered in a series of its decreasing size, largest chromosome at the first and smallest at last. So when a karyogram is made, it is basically made in a descending order. That is, the largest chromosomes are placed first, and slowly as the size of the chromosomes decreases, so it is numbered as accordingly. In the case of humans, the autosomes are numbered from 1 to 22 and arranged in the order of decreasing size. Sex chromosomes are arranged after the autosomes. Chromosomes in the karyogram are aligned along a horizontal axis shared by their centromeres. The crucial step is the isolating and culturing living cells. The entire karyotyping process begins with a viable biological sample. For a routine postnatal or adult analysis, the most common sample is a peripheral blood draw collecting in a sodium heparin, that is a green top tube, anticoagulant tube. For prenatal diagnosis, the sample may be amniotic fluid containing fetal amniocytes or chorionic villus samples, CVS. In cancer diagnosis, it is often a bone marrow aspirate or a piece of solid tumor. So these are different, and the anticoagulant vial, which is used for collection of the sample, blood sample, is sodium heparin, which have a green top cap, and the different samples which are used for the diagnosis for different type of disorders. But a critical problem must be overcome. Most of the cells in these samples are not actively dividing. The T lymphocytes in the blood, for example, are in resting non-dividing state known as the GO phase of the cell cycle. This poses a problem because chromosomes in their familiar X shape only exist during the mitosis, that is the cell division. In a resting cell, the DNA is uncoiled into a long tangled mass called chromatin, making it impossible to analyze. So it has to be it should be put in a dividing stage so that the chromosomes can easily be identified, and they come in in a condensed manner, so that it becomes convenient to visualize them. So the first step is to move or shift from the resting stage to the dividing stage. This is why cell culture is needed in karyotyping. We cannot analyze cells that are not dividing. Therefore, the first step is to bring these cells to life in the laboratory setting. The process of cell culture chromosome analysis involves several key steps. First is that we have to culture the sample and add some of the important nutrients for it, or excite it to in a dividing stage. And what are some of the important steps? That is the aseptic. The sample is handled in a sterile biological safety cabinet to prevent contamination from bacteria or fungi, which will quickly overrun the culture. Inoculation, a small amount of the sample, for example, blood, is added to flask or tube containing sterile nutrient-rich broth called a culture medium. The medium used for this is RPMI 1640, which contains essential nutrients, growth factors, and stimulants, which helps to it to shift from the resting stage to the growing stage and dividing stage. This medium contains salt, sugars, amino acids, and other essential building blocks for cell growth. Stimulation, that is the mitogen, to kick-start the resting T lymphocyte into dividing a mitogen is added. The most common one is 5-hemagglutinin, PHA, plant-based lectin that stimulate T cell proliferation. This is the on switch for the cell culture. Incubation, the flask is placed in a specialized incubator, a warm 37 degrees centigrade, or we can say 98.6 degree Fahrenheit humidified environment with a controlled atmosphere, typically 5% carbon dioxide, that mimics the condition inside the human body. So we have to give a condition which is similar to the human body, and that's why these conditions are added artificially to shift the cells from the resting stage to the dividing stage. The cells are left to grow and divide for 48 to 72 hours for blood, or even 7 to 10 days for amniocytes, which grow more slowly. During this time, the single T cell we started with multiplies into a large population of effectively dividing cells, providing the raw material for the next phase. Karyotypes are prepared from mitotic cells that have been arrested in the metaphase or pro-metaphase portion of the cell cycle, when chromosomes assume their most condensed conformations. A variety of tissue types can be used as a source of these cells for cancer diagnosis. Typical specimens include tumor biopsies or bone marrow samples. For other diagnosis, karyotypes are often generated from peripheral blood specimens or skin biopsy. For prenatal diagnosis, amniotic fluid or chorionic villus specimens are used as a source of cells. The process of generating a karyotype begins with the short-term culture of cells derived from a specimen. After a period of cell growth and multiplication, dividing cells are arrested in metaphase by addition of colchicine, as we have already discussed earlier, which poisons the mitotic spindle, which prevents, we can say that it prevents the formation of spindle fibers, and the cells are next treated with hypotonic solution that causes their nuclei to swell and the cells to burst. So as the cells burst, chromosomes or nucleic acid part comes out, and these are then used for the analysis. So the nuclei are then treated with a chemical fixative, dropped on a glass slide, and treated with various stains that reveal structural features of the chromosome. So through this diagrammatic representation, you can see that here the blood has been withdrawn and it has been cultured for 72 hours. Basically, blood samples requires 42 to 48 to 72 hours to enter into the dividing stage, or we can say metaphase stage. Then colcimide, that is the colchicine, to arrest cell division at metaphase. Further centrifugation is done, and then hypotonic solution to rupture the red blood cells and swell inside blood cells. Further centrifugation is done, and methanol and acetic acid to fix cells is added so that they are fixed. Then white blood cells in fixative. Then place a few drops on a slide, dried, stained, and analyze the karyotyping. So these are some of the important basic steps, and you can see here under the microscope, we can see the different pairs of chromosomes, their structures, structural and number easily we can identify. Now coming to the next, that is the how we stain these chromosomes. So though the staining chromosomes is called as the banding techniques, and there are different types of banding techniques. The banding patterns rely on differences in DNA density and composition along the chromosomes. So there are two portions. There is one is heterochromatin, which are tightly packed, really replicating DNA, often AT stays dark with GC banding, and G banding or euchromatin, loosely packed, early replicating DNA, often GCH, which stays with G banding. That is, they are light bands. Pre-treatment before staining slides are often treated with heat enzymes like trypsin or acids to denature proteins and differentially expose DNA. So any protein structures which are present along with the DNA, basically histone proteins and other proteins which are attached to the DNA are removed by the enzymes like trypsins or acids to denature them, and that exposes DNA. So any other materials or any other substances which are present in the DNA are first of all removed by pre-treatment. So what is the procedure for gDNA? So age the air-dried metaphase slides overnight in a drying oven at 55 to 60 degrees centigrade. Remove and bring the slides at room temperature. Dispense 50 ml trypsin solution, 50 ml of 1% fetal bovine solution, 50 ml of 1x Giemsa stain solution, and 50 ml GER buffer in 4 Coplin jars. See, these are the different, that is the 50 ml trypsin in A, 50 ml of 1% fetal bovine solution in Coplin jar B, 1x Giemsa stain solution, Coplin jar C, and GER buffer in Coplin jar D. For processes that hold the slides with the help of forceps immersed in a coffin jar containing a trypsin solution exactly for 8 to 10 seconds, moving the slide back and forth. The exposure time to trypsin should be adjusted depending on the quality of the cytogenetic specimen and the result banding. Briefly rinse the slide in a Coplin jar containing 1% fetal bovine syrup to inactivate the trypsin. So after the fetal bovine serum, remove the extra DNA parts, extra nuclear parts. Then their activity has to be inactivated, and that is done in the Coplin jar B, which contains 1% FBS. Then rinse the slide by dipping in a Coplin jar containing GER buffer using the same agitation technique as in the step 3. Place the slide in a Coplin jar containing Giemsa stain solution for 8 to 10 minutes. So this is the staining, or that is the Giemsa. Now rinse the slide in sterile distilled water. Giemsa is a type of Romanowski stain and is a type of differential staining methods containing methylene blue and eosine. Rinse the slide in sterile distilled water until the stain no longer comes until the stain no longer comes out, and then and until this Stain no longer comes out, and then we have to remove the slide. Long exposure to water will result in de-staining the slide. Allow the slide to air dry and examine by light microscopy using a phase contrast microscope to determine the quality of banding. Adjust trypsin exposure or duration of staining as required. Once optimal banding quality has been achieved, analyze the slides, store the slides in a slide box free of dust and dirt at room temperature. You can easily visualize or see here that how the banding patterns are seen after staining with Giemsa stain. So here other stains are Q banding, which is used instead of Giemsa stain. Quinacridine stain is used. That is quinacridine dihydrochloride or quinacridine mustard, and it's the simplest and the first chromosomal banding method. Quinacridine stain chromosomes show a characteristic pattern of bright bands on a darker background. Because quinacridine is a DNA intercalating agent and a fluorescent compound, the bands appear only when the chromosomes are exposed to ultraviolet light. Usually, for this, we have to use fluorescent microscope, but as in G banding, we have to use phase contrast microscope. But for a Q banding, as it is only under the UV light, it will fluoresce. So we have to use a fluorescent type of microscope. The only difference is here of chemical, that is their Giemsa stain is used and here quinacridine stain. Then is C banding is a centromeric heterochromatic staining. Heterochromatin staining. In this technique, before using Giemsa stain, the cell is pre-treated with alkali. Therefore, this technique is also known as CBG staining, B banding by base with Giemsa. Centromere heterochromatin, ancestral part of Y chromosome containing highly repetitive DNA sequence is used to stain by C banding technique. After staining, banding is viewed by bright field microscope or banding. So different here, we can see that before using Giemsa stain in C banding, the cell is pre-treated with an alkali. And so this is also known as CBG staining, and this is basically viewed under bright field microscope. R banding is called as reverse chromosomal banding. What happens here that the dark band in the G banding, the heterochromatin region shows dark band, and euchromatin region shows the light band. But here the technique here it is reversed. That is the dark band, that is the 80 region observed in G banding technique appears light in R banding, and light bands, that is the GC region or euchromatin region, appears as dark. So here also basically we can see that all uses C banding, R banding, Giemsa stain, but some of the technique varies which distinguishes it from the G banding technique. Then next is the chromosomal painting. It is the most advanced technique. With this technique, colorful chromosome images are created by treating chromosome spreads with fluorescently labeled DNA fragments that have been isolated and characterized in the laboratory. At first, the DNA fragments isolated from desired gene is labeled with fluorescent dye and is applied to chromosome spread on a slide. The fluorescent labeled DNA fragment will bind to chromosomal DNA that is complementary to its sequence, and this binding results in visual colors at the position of complementary sequence. So it is a type of DNA probe assay, and a type of DNA probe where a fluorescent labeled probe is used to identify the desired chromosomal structures, and it is visualized through complementary base pairing. So this is one of the important new modern technique. Now, what are the uses of karyotyping test? The karyotype test has several important uses in both clinical and genetic settings. Genetic disease diagnosis help diagnose chromosomal abnormalities, including conditions like Down syndrome, Turner and Klinefelter syndrome. Prenatal testing detects chromosomal abnormalities in the fetus, such as trisomy 21, as well as other genetic disorders. Cancer diagnosis identifies genetic changes associated with cancers, particularly leukemia and lymphomas. Infertility investigations helps identify chromosomal abnormalities that may affect fertility or pregnancy outcomes. Developmental delays used to assess developmental delays in children and diagnose any underlying genetic conditions. So, in to summarize, it is one of the most important technique, and it is one of the most advanced or modified technique on which cells are visualized at the chromosomal stages. We know that usually cells, when we identify cells through different staining techniques or other methods, we usually see the structure and morphology of cells. But some of the important underlying abnormalities cannot be detected through cell morphology or study of the cells. That is the basically cytology. So here we go at the genetic level because we know that most of the important proteins or enzymes or any type of the structures which are present in our body is regulated on the level of genes or genetic coding. So it becomes easier for us to detect all those abnormalities or any abnormalities at the structural level, which either overexpresses some of the genes or it will suppress at the chromosomal level. So it becomes a very important, it is a very important tool in the modern day medical sciences, and it is quite useful for diagnosis of some of the important syndromes or disorders which cannot be or which is very difficult to diagnose at the cellular level. So with this, I conclude my today's lecture. I hope you have got some benefit from my lecture. If you like it, please do like and subscribe to my channel, and it will motivate me to make some more valuable lectures for you. Thank you.